So good evening, everybody. We're so glad that you could join us today. I hope you're all doing well and are staying safe. My name is Saloni Haldule, and I am a third MBBS student at B.J. Medical College Pune, and I'm also the head of the networking team at the Jignyasa Research Club. I will be your host for this evening. Joining us today is Dr. Dhanasekaran Shanmugam. Dr. Shanmugam is a senior principal scientist at the National Chemical Lab here in Pune, and he is also an associate professor at the Academy of Scientific and Innovative Research in Chennai. Sir was a postdoctoral fellow at the University of Pennsylvania in Philadelphia, which is a prestigious Ivy League research university in the US. He has received many awards for his work, including the Giri Medal for the best thesis from the Department of Biotechnology in the Indian Institute of Science in Bangalore, among many other awards. He is also a member of the Scientific Advisory Committee on the National Institute of Malaria Research, and has 33 peer-reviewed articles published in various journals in his field. Welcome, sir. It's an honor to have you here with us today.
So first, thank you for inviting. Let's get started. Yeah. So once again, welcome everybody, and thank you all for coming to this Jignyasa Club talk session. And I would like to thank the students of B.J.Govt.Medical College. This is a wonderful thing that they're doing, inviting scientists from national laboratories to talk to college students and medical school students. And today my topic of discussion is how genomics can help malaria elimination. So let's just get started here. So this malaria parasite belongs to a group of unicellular eukaryotic organisms. Collectively, these are called epicomplexan parasites. They belong to this phylum Epicomplexa, and Epicomplexa is a very large taxonomic collection of organisms, many thousands of them, and all of them are parasites. And many of these parasites cause different kinds of diseases in humans and animals. For example, there is another parasite which causes toxoplasmosis. It's called Toxoplasma gondii. Some of you might know it. And malaria is caused by Plasmodium species. In human, there are at least, so far known, at least 5 different types of Plasmodium species which can cause, but the most dangerous is Plasmodium falciparum. And some of you would have studied this in college. Anyway, it's interesting that there are many such parasites causing diseases, and many of these parasites, it's difficult to study them in the lab. But genomics is helping us to understand these parasites much better, even without being able to, some of them cannot be cultivated in the lab, or we cannot perform experiments with them in the lab, but still we are understanding their biology from a genomics perspective. And I'll tell you in this talk how that's possible. So the important information we have to understand is, for many of these diseases, we only have very few drugs available. And these drugs are sometimes useful, but many times not useful, because the parasites have become resistant to the drugs. And right now, if you take malaria, the most commonly used drug is this artemisinin or its derivatives. And artemisinin alone is not used. It is used as a combination with some other drug. And this artemisinin combination therapy is the first-line treatment for malaria. But already reports from Southeast Asian countries like Thailand, Myanmar, and even some parts of India have reported artemisinin resistance in malaria. So it's very critical that we discover newer drugs so that we can keep fighting against these diseases. So I'll tell you how genomics is going to help in this fight against these parasites. Okay. So before we go into the genomics part, I just want to show the life cycle of the parasite a bit. You focus only on the left side of the screen, which basically is the malaria parasite. So the malaria parasite is transmitted by mosquitoes, and only certain species of mosquitoes. In this case, it will be Anopheles species of mosquitoes. And wherever Anopheles species mosquitoes are endemic, then you will have malaria transmission. So usually it's in the tropical regions of the globe. So when the mosquito bites a human, actually it injects the sporozoites. These sporozoites are a stage of parasites which are capable of infecting the human. They go to the liver, develop there. Then from the liver, they go to the blood. And you can see that the blood cells are there. They're infecting the blood cells. And then that is when the disease manifests, when the blood cells are getting lysed. And then there is huge hemolytic shock in the host. But some of these parasites can also differentiate into male and female gametocytes, and then they get picked up by a mosquito once again. And these gametocytes can combine inside the mosquito to form ookinete stage. And this then develops again back into sporozoites. So this completes the cycle. So what I'm going to quickly do now is there are a couple of small short videos which really highlight the life cycle of these parasites. I thought this would be very nice to share with you, so I'm just going to show you now. So this is the life cycle of the parasite as it develops in the mosquito. What happens in the mosquito? So by the way, these videos are put out by the Harvard Hughes Medical Institute from USA, and these are online in YouTube. You could as well see it later on. I've started the video now. Just let me know if everything is going well and you can hear it.
Yes, sir. I'll.
Vegetarian, preferring to drink nectar, fruit juices, and honeydew. Only a pregnant mosquito will bite humans, seeking nutrients.
Sir, I think your mic has been muted. Can you just check your mic on Google Meet? Has been muted. Can you just unmute it?
Yeah, I am muted. Now you can hear me?
The tiny drop of blood filling the
I
insect's
don't know what
stomach.
happened. Now everything is fine.
Parasites.
Everything is fine now?
Yes, sir. Yes.
The parasite form that is deadly inside humans cannot survive in a mosquito's stomach and is slowly digested with the rest of her blood meal. However, back in the human host, a few of the parasites turned into a different type of cell, one that is sexual but remains dormant. Malaria sex is triggered when the warm human blood begins to cool inside the insect's stomach. The female form of the parasite matures into an egg. The male form takes a while longer to mature into sperm. This sperm is from an earlier feed. The fertilized cell can glide and begins to explore its new environment. It migrates to the outer lining of the mosquito's stomach before transforming into a cyst. Each cyst produces thousands of thin, tiny parasites which seek out and infest the mosquito's salivary glands. The next time this mosquito bites a victim, the malaria parasite will ride in with her saliva and infect another human. This year, 10% of people on Earth will be struck down with malaria. Most people who die from the disease will be pregnant women and children under the age of 5.
So what you just saw was how the parasite develops within the mosquito. Now the mosquito has bitten a human, and we can now see here how the parasites develop within the human host. So I'm just starting
The
this
malaria
video.
parasite is an ancient organism. It has been with us since before we were human. Famous victims include Alexander the Great, Genghis Khan, and George Washington. The malaria life cycle follows a devious path, swapping back and forth between mosquitoes and humans. This mosquito is infected with the malaria parasite. Because she is pregnant, she has become hungry for human blood. During the bite, she injects saliva to stop the blood from clotting. Her infected saliva also carries the malaria parasite. The parasite rides the bloodstream like a network of roads, seeking its first target, the core of your body's blood filter system, the liver. Sensing its arrival at the liver, the parasite searches for an exit. A sentinel Kupfer cell is the entry point to liver tissue. Leaving the blood, the parasite infects a liver cell, killing one or more other cells on its way. Over the next few days, the parasite undergoes hundreds of nuclear divisions, copying its DNA over and over again. A single infected liver cell can create thousands of new parasites. The next generation of parasites are modified to infect a new target. Red blood cells. Inside a red blood cell, the parasite can hide from the body's immune system. The parasite slowly devours the contents of the infected cell and creates more parasites. The infected cell becomes sticky and grips onto blood vessel walls. Once mature, the infected cell bursts, spreading more parasites through the bloodstream. Malaria victims suffer fever, loss of blood, convulsions, brain damage, and coma. This year, 10% of people on Earth will be struck down with malaria. Countless millions have been killed by it. Most people who die from the disease are pregnant women and children under the age of 5.
Yeah. So that's how the parasites develop. The malaria parasite develop in the human host as well as the mosquito host, and really understanding the biology of this parasite has taken more than 100 years of work now. Malaria was always there with human beings for many thousands of years, but only in the past 100 years, the parasites have been identified and how they are transmitted by mosquitoes have been identified. And today we have all the molecular biology tools to study this parasite. Like I said, the most important focus now in malaria is to discover new drugs. So in my laboratory, we do a lot of screening to identify molecules which can kill the parasite. So the important thing is, as you saw in the life cycle video, when the parasites infect the red cells, they cause lysis of red cells. And this lysis actually results in hemolytic shock and immune response and everything else. And that's why you get fever and all the associated disease symptoms. So we would like to have molecules which can arrest the development of the parasite inside the red cell and kill it. So we do that. We can grow these parasites in the lab, and as you see in this scheme here, we take the parasites, we add some chemicals, and we see whether any of the chemicals can actually inhibit the development of the parasite from one stage to another stage. And we have done many such screens, and we have screened, in fact, many couple of thousands of molecules. We have very good inhibitors now, but these are all molecules which are still in the lab. For these molecules to actually become medicine, it has to go through a very lengthy drug discovery process, and it's time consuming, it's very costly, but we have to do it. And so what right now we are doing is we are taking these molecules and going to preclinical studies, which is basically testing them in animal models for malaria to see whether they're efficient in killing the parasite and curing malaria in animals. And then we have to study the pharmacology of these molecules, whether they are toxic to humans, whether their half-life in the blood is good enough to kill the parasites, and so on and so forth. So that is why it's a very lengthy process. But today I'm not going to spend much time talking about drug discovery. I will tell you how genomics is helping in understanding parasite biology. So around the 15 years ago, the genome of the parasite was decoded. Okay, I hope you all understand what the genome is. It is the nucleic acid which is in the nucleus of the parasite, which codes for genes and so on and so forth. And this genome has all the information of the parasites. What genes are expressed, and what kind of genes, what kind of proteins are produced by the parasite, and what can be targeted. Of course, because we are getting a huge amount of this genome data, we would like to have an easy way to analyze this data. So I'm again going to show you something now from a website, which is basically Plasmodium genome resource. Okay, fine. So this PlasmoDB has all the genomics data, and you could actually go and you could get all the information about how many genes the parasite has, what type of proteins are coded, and so on and so forth. So it's a very extensively interactive database. Unfortunately, we won't have time to go through all of these things here. Many of you may not be knowing how to do genomics analysis, so I'm trying to keep it simple here, but later on in question and answer session, if you want something, you can talk, we can discuss about that. I just want to show you one simple example here. I just try to do some search analysis. So for example, I showed you these parasites, they grow within the red blood cells. So I want to know what, when the parasite have to go into mosquito, they must become the gametocyte. So there are certain genes which are expressed during the gametocyte stage so that it helps in mosquito development. So now I can go and look at what are those genes. So I'm going to this evidence for gene expression and just keep things simple. I'm going to go with some microarray data. Don't worry about what you're seeing here. I'll just quickly show you something you will understand in a minute. So these are different experiments, and it doesn't matter which experiment you choose. They are slightly different from each other. So you can choose whichever experiment you think is good enough for your analysis. So I have chosen this experiment here, and I'm just clicking on this percentile here. So I'm just going to select gametocytes, because these gametocytes are the one that can survive in the mosquito and develop into sporozoites, which can again reinfect a human being. So I don't want gametocytes to form. I want to kill them. So I want to find out which genes are important. And so I select the gametocytes, look for gene expression, and then I try to get the answer. So I've done this kind of analysis already, and we can see here that there are almost 1,226 genes which are highly expressed in gametocytes. So now, where are these genes located in the genome? So I can go to this Genomics View. So what you are seeing here actually is all the chromosomes of the parasite, just like how human beings have 23 pairs of chromosomes. We have the malaria parasite has 14 pairs of chromosome, not 14 pairs, it is 14 chromosomes. These are haploid. Of course, they become diploid when they are when the gametes mate and make the ookinete. But otherwise, in the human beings, it is only a haploid. So 14 chromosomes, and you can see the genes are all located in different chromosomes here. So this is just a genomic view. But you can go to any one of these genes, and you can get, so this is the gene details. So I'm just showing you how you can get details of each and every gene. So there are many different data types, like for example, protein expression and properties and so on. And you can go to any of these data types. It's buffering here a little bit. Just a minute. Okay, it's not loading. Okay, let's try something else. So we can also look at the genomic view. So basically what you are seeing here is the genomic view, and this protein is called histone H2B. It's a histone protein, which is a DNA binding protein, very important for condensing DNA and keeping it in the nucleus. So here is that gene, which I'm seeing, the histone H2B gene. But then I want to see what are the neighboring genes, and whether it is interacting with any other gene. If I want to design some experiment, I should be able to understand its genomic location, whether it has mutations, and so on and so forth. So to do all of this genomic view and genome browsers are very important. Again, there is a lot of details here. I'm just showing this to you for just a quick appreciation of what can be done. So now, if we go back to the PowerPoint slides. Okay. All right. So I'm just going to skip a couple of slides here. I'll just go to this slide here. So here it is a summary of the number of genes in the Plasmodium parasite. You can see here it is highlighted. So the Plasmodium parasite encodes nearly 6,000 genes, and about half of these genes are actually of unknown function. That means we don't know what these genes do, and these are all genes which can be potentially targets for drug discovery. So how do you understand the function of these genes? So a lot of bioinformatics can be done to check what the function of these genes can be. So this is one of the things we do in my lab, which is we identify all the enzymes that are participating in metabolic pathways. So I hope you all understand that every cell has lots of different kinds of metabolic pathways, and all of these pathways are important for the cell to live. So if we can understand which metabolic pathways are very important for the parasites to survive, then we can target that enzymatic function with some drugs. and to identify the complete set of enzymes so that we can make this kind of metabolic network charts. We need to have genome information. So from the genome, we can identify all the genes, and then we can find out which are the genes encoding the enzymes and what type of enzymes are encoded. And once we know what type of enzymes are encoded, we can make this kind of networks. And from this network, we can identify what is the weak point. Where is the parasite having insufficient backup in terms of metabolic capability? And so those weak points we can target with drugs. Of course, it is not as easy as I'm making it sound. It is very challenging, but this is one of the way we get started into genomics here. And from that kind of a network, we can actually make this kind of tables. And you can see here, I have compared metabolic pathways of different parasites with humans. And anything that is highlighted in red here are all unique to the parasite, which means you can see that, for example, fatty acid biosynthesis, humans have type 1, but many of the parasites have type 2, including the Plasmodium malaria parasite. So this is a different way they make fatty acid biosynthesis. So if we target this function with some drug, it will not be toxic to the humans because humans have a different way of making fatty acids. So we need to understand what are the differences and similarities between human metabolic pathways, parasite metabolic pathways, and what can and cannot be targeted for drug discovery. So while doing all of this, there is, well, I just have to take a step back here, and I want to introduce something. So WHO, which is, you all know, is the World Health Organization. It is the premier global organization that coordinates health response. And nowadays, because of the pandemic situation, everybody has heard about WHO and what they do. So WHO has many sort of subsidiaries also. So there are subsidiaries like TDR, which is Tropical Disease Research. And then there is another subsidiary called MMV, which is Medicines for Malaria Venture. And I'll show you information about that in a minute. So these organizations, actually, they fund different groups and labs for infectious disease work. And one of the projects we had was how we can use genomics data sets to identify drug targets and then use the drug targets to make new drugs and so on and so forth. And I just want to show you another database, which is this TDR targets database, where one can go and actually look at all the genes in the parasites, and you can ask questions like, you can select the genes based on some user-defined criteria. So this criteria are nothing but just questions. So if you want to say that the gene must encode for an enzyme, and the enzyme must not be present in humans, it should be present only in parasites. So if you have all these questions, then you can filter and you can select a few target genes in this database. And then this is just the list of criteria we can use to select such targets. And then we have done some exercise. We have published some of these papers as well. This review, Nature reviews, we published a while ago. So you can see how there are, if you take the class enzymes, which is the first entry in the table here shown, we can identify almost 850 or so enzymes in Plasmodium. So if a biochemist is interested in characterizing novel enzymes or unique enzymes, then they will focus only on these genes because these are only the enzymes. They will not worry about the other genes. But then the idea behind this filtering criteria is that you want to have enzymes, and enzymes should be, we should be able to make the enzyme in the lab. We should be able to characterize its function in the lab. It will be nice if there are crystal structures for these enzymes. And so we can add multiple criteria, and then we can come up with a list of genes which satisfy all this criteria, and then we can take these genes forward to do further analysis. And you know, by doing this kind of analysis, we get a lot of information about the function of the gene. Sometimes the function of the gene is not known in the parasite. So what we do is, if the function of the gene is known from another organism like bacteria or yeast, or even in human beings like, you know, orthologs and so on and so forth. So basically, we try to take functional information even from other organisms, because as I told you at the beginning of my talk, some of these parasites we cannot grow in the lab. We cannot really study their biological properties. You know, we cannot purify this enzyme. So it will be very difficult to do experimental work with some of these parasites. But if the same enzyme is present in, say, bacteria, which we can grow in the lab very easily, we can take the information available for the bacterial enzyme and we can apply it to the parasite enzyme and say, okay, both look very similar. So how do we maximize the data from bacterial enzyme to parasite enzyme in terms of drug discovery? So those are the kind of things we ask, the questions we ask. So don't worry about this slide, this technique of orthology-based mapping. I understand that many of you may not be very familiar with the genomics terminology and genomics background, but of course we can discuss later on in question and answer sessions. So I'm just going to skip a few things here, go back to, okay, so here I just want to show you. So if you want to have more detailed information on malaria, this is a very good website which is called Medicines for Malaria Venture. And here all the current updates on malaria is given. Of course, Medicines for Malaria Venture deals with a couple of other diseases as well. Here you can see something called COVID box. Let's not worry about what it is now. But if you want to look at more details on malaria, it is here in this link. You can go here and you can actually see what are all the medicines, new medicines which are in the pipeline. So I can just show you some details here. Like for example, okay. I think I'm just trying to see. So I'm just trying to find a table which gives all the new medicines which are getting developed for malaria. I'm just not able to find that table now. But just give me a second here. So this is the table I was looking for. So this gives you an idea of the difficulty in developing medicines, not just for malaria, but for any disease. So everything has to start in the lab where research is done. And many times it's not just institutes like mine which are doing research in discovering new medicines, but many of the companies like GSK, Novartis, and there are many other pharma companies which invest in this process. So once a lead compound a lead chemical compound is identified, that chemical compound is taken forward for various levels of testing, like candidate profiling, preclinical testing, and then clinical trials begin, where human volunteers are tested, and then when all the pharmacological parameters are satisfactory, then they go into clinical phase 3 and 4 where real patients, malaria patients are tested. And then final round of regulation happens and then approval comes. So luckily for the past, couple of decades, sustained efforts in malaria drug discovery has resulted in identification of many different types of molecules. These molecules are now approved for, they're not approved for coming to the market and being prescribed in hospitals for patients, but these are all approved for the final stages of regulatory process. And then some of them are older drugs in newer combinations, but some of them are newer, completely new drugs as well. The most recent drug which made a lot of news for malaria was tafenoquine. Actually, there is a very interesting fact that I told you Plasmodium falciparum causes severe malaria in humans, which can kill. There is another parasite called Plasmodium vivax. And this vivax malaria, it doesn't kill, but it is very debilitating. So it is equally important to address this disease. And in India, Plasmodium vivax is very prevalent. Most of the malaria is caused by this vivax parasite. And nowadays, what is happening, we are seeing even with vivax malaria, many people are becoming severely ill and some of them are even dying. So the parasite is also, looks like it is changing a bit. Again, genomics can help us understand what kind of changes are happening in the parasite. But this tafenoquine has been approved specifically to treat vivax malaria. And one of the interesting things with this vivax is, after treatment, the patient feels that he is completely cured and there are no parasites in the blood, but some residual parasites are still sitting in the liver. And so many months or sometimes even a couple of years later, there can be a relapse of the disease. So the parasites which are sitting in the liver dormantly can once again come back and infect. So this tafenoquine medicine actually, it takes care of those dormant parasites. So this is a very real important medicine which has become available for malaria, especially in the context of malaria in India. So there is a lot of information we can get out of genomics and how it can help in malaria drug discovery. So my intention was just to give some kind of an overview. Of course, we didn't go in depth into any one topic, but I can stop here and take questions.
Yes, sir. So there are questions that have come. Okay. So the question that we got was, what is the exact role of hydroxychloroquine in treating malaria, and what is in what way is it helpful in treating the COVID-19 illness?
So why am I not surprised by this question? Anyway, first things first. Hydroxychloroquine by itself is not a malaria treatment. Okay. There is another drug called chloroquine, just simple chloroquine, which is what is used for malaria treatment. Now, this hydroxychloroquine also can kill malaria parasite, but it is not as good as chloroquine. So people don't use hydroxychloroquine for malaria treatment. Where hydroxychloroquine is used is in this condition called lupus, systemic erytheromatous lupus, I think. And then actually hydroxychloroquine has some kind of an anti-inflammatory effect on humans as well. So any drug can have different effects on different organisms. So this drug, it kills malaria parasite, but not as good as chloroquine. But at the same time, it has some effect on the human immune system, which helps in some other disease. So basically, hydroxychloroquine is it's used in arthritis and lupus and things like that, and it's widely prescribed and used, quite safe to use in that way. So somehow in this COVID situation, again, I am not a specialist in COVID or I'm not a medical doctor by profession, so I'll take my comments as just comments only, and these are not facts, and nobody should quote me on anything here. But I'm just saying that I think what is happening is hydroxychloroquine is not directly helping kill the virus, and now we have seen some clinical trial data published. There was some controversy in some data that got published, but I think it's pretty much clear. I've been following some of the literature, published literature, scientific literature. What is happening is it is acting through the human immune system. And in COVID, one of the reasons people die in COVID infection is it's not, of course, the virus is multiplying in the person, it is a disease burden, but why is one person able to be cured and come out of the disease while the other person is just succumbing to the disease and dying? I think it is because the way the immune systems react. So one person's immune system is able to control the virus as well as it is not overreacting to the virus. But another person's immune system is just overreacting to the virus, and hyperinflammation can result in death. And as medical students, many of you will know that. And I think hydroxychloroquine helps to dampen that inflammatory response to some extent, but it is not very good at that either. So I think there is now some level of evidence for that, and even WHO has now recently recommended that hydroxychloroquine is not very useful for COVID treatments.
Okay, sir. Thank you for that. Now, we have a question for your about your research journey. So, as we know, we're all students that are interested in research, and especially medical research. So, people wanted to know how did you develop an interest in the field of research, and what were some of the obstacles that you faced that you had to overcome?
Yeah, I mean, this interest in in research, or in particular disease research, it just didn't happen overnight. Actually, I for my bachelor degrees I did biochemistry, and when I was reading biochemistry, I got very interested in metabolism. So my first interest, if you will, was on metabolism and not on disease research per se. But then for my master degree, I went to Jignyasa Medical College in Pondicherry. I am a native of Pondicherry, so I went there to do my master's in clinical biochemistry. And that is where I got exposed to, more exposed to disease biology. And I thought, okay, I'm doing biochemistry, so how I can apply biochemical insights into disease. And then when I went to my PhD, that I just happened to get the opportunity to join a very good lab which was working in malaria biology, and just things fell in place there. Of course, my research interest was mine, but going particularly into malaria happened only because I could get the opportunity at the time of doing my PhD to join the lab. And obstacles wise, of course, as with everybody, getting into a good college or getting into a good institute is a big bottleneck, and you have to really work hard to do that. And I was lucky in many ways. I could get to do my PhD in one of the best institutes in India, which is Indian Institute of Science, under very good mentorship of two guides, Professor Padmanabhan and Professor Rangarajan. And so I was lucky in many ways. But the obstacle part was that, of course, there was when I joined for PhD, there was no genome of the parasite. We didn't know anything about the parasite. What we knew about the parasite was all basically biochemical experiments and some level of cell biology experiments. And I can say that the first couple of years of my PhD, I had done much of the work in the dark, I would say, because of lack of genome information. But once the genome became available, we could clone many genes, we could characterize many genes. Things moved very fast. So basically, rather than saying that it was a big obstacle for me, I can say that, of course, at the beginning it was an obstacle, but having genomics towards the later part of the PhD work became a very a big blessing for me, I would say.
Okay, thank you.
Yeah.
So another question that we've got is, what do you think is the scope for biotechnology research in India? And what is the difference in research overall between India and the US?
I think this is in a way, this is a little bit of a vague question, because I think if any more specifics can be there, it would be good, because biotechnology is such a vast field. There is agri-biotechnology, there is marine biotechnology, there is medical biotechnology. So, I mean, if you want to comment on biotechnology as a whole, that will be very unfair, because every field of specialization has different levels of biotechnological advancements. Where I would say really India is doing well, let's just focus on, say, medical field for the time being. And then I would say that in recent years, many of you would have known that antibody-based therapies are becoming a big thing in not only in cancer, but in many other areas. In fact, just a couple of months before, there was a very good Nature paper published where people have identified an antibody which can kill the malaria parasite very quickly and very potently. So in addition to chemicals being used as drugs or medicines, we are going to have proteins or biologicals being used as drugs, and those are the future. And in India, there are many companies now, biotechnology companies, which are investing in this biologics. And I think that is a wonderful thing. That is a big thing happening in India. And in India, as many of you know, we are also one of the world's biggest vaccine producers for viruses and various other diseases. Even for COVID, there is a lot of talk of Indian companies partnering with, other international companies to come up with the vaccine production and things like that. In Pune, there is the Serum Institute. Some of you might know. It's one of the biggest vaccine producers in the world. If any of you get an opportunity to visit that institute, you should go. It's basically a company. They call it Serum Institute, but you should go there and you should see the kind of facilities there. It's very inspiring, because what we do in the lab in small scale or what we read in college and textbooks is very different from practically when you set up a big industry, like a big plant to produce millions of doses of vaccine. It's a big challenge, and these people are doing it. So I think we are in very good, there's a very good trend in India in terms of biologicals and vaccines in the area of biotechnology. I think agri-biotechnology, marine biotechnology, and animal biotechnology, it's again, it's a whole different universe there. I'm not an expert in those areas, but I can tell you that, agri-biotechnology is again doing very well, because we have developed new varieties of crops which are being tested now. But again, there is this whole problem of genetically modified organisms. Can it be used as food, or can it be used as plant-based or animal-based food? Can they be used? So those are all policy decisions, but as scientists, people are developing these things, and sooner or later, I think they are going to be very useful to cater to the needs of the nation.
So, sir, since you mentioned vaccines, how likely do you think it would be that in this generation we'd see a vaccine for malaria?
Well, vaccine for malaria has already been, so I should say that that is one of the sort of the holy grail in malaria, is to discover a vaccine. And so different approaches have been taken. Okay, so from what I know scientifically and what is there in the public knowledge. So if you look at malaria vaccine, there is, along with the WHO, some company has developed a vaccine already, GSK. So they have developed this vaccine. Initially, it was developed by another company, and then GSK took over. And also, anyway, so this vaccine has been tested in Africa. By the way, Africa continent has the highest malaria burden. the world, and more children die there than anywhere else in the world due to malaria. So most of the drugs and vaccines by default get tested first in Africa before being sent to other places. So this vaccine was tested in Africa, and then it gave a protection level of around 30%. So it's not 100% protection. So we have a vaccine, but it's not very good. So the question is whether we can get a vaccine like a polio vaccine, which is 100%. Like if you take the polio vaccine doses properly, you are 100% protected. You can never say that for malaria. It is only 30% protection. So what is the challenge? Why can't we make a vaccine for malaria? The challenge is again in the genes. This is where again genomics can help, which is, you all know that genes can get mutated. The sequence of the genes, the nucleotide sequence of the genes can change. And sometimes they change naturally. Every organism changes its DNA. Even humans change their DNA. Whatever DNA we are born with, we change it continuously. And if you check any one organism's DNA over a period of time, you will see changes. But those are natural changes and natural mutations, and some of them get corrected by the cell, some of them are not corrected. And many of these mutations have no big effect on the biology of the organism. But every once in a while, a mutation can actually be a big advantage for the parasite. So when the parasite, especially the malaria parasite, it looks like it can mutate its genes very easily. And that is why it is able to become resistant to many of the drugs. One of the problems with vaccine is also that whichever protein you use as a candidate vaccine, so that protein sequence is not the same over a period of time, and it can change. And there is also a lot of immunology from the human side which affects how effective the vaccine is. But the parasites are also capable of changing their protein sequences, and so one vaccine which is effective today may not be effective a few years later. So that is one of the big challenges. And in previous years, people have also tried using dead parasites to immunize and see whether it can protect. Actually, dead parasites protect very well, but somehow getting an approval for using a dead parasite as a vaccine is a big bottleneck again. What if one parasite is alive? Like you are killing thousands of parasites and making it into an injection that can be injected into you. But somehow if one parasite escapes being killed, it can cause the disease. So that risk is there. But otherwise, there are other technical factors also which inhibit killed parasites as becoming vaccines. But by and far in research, what we have seen is when you inoculate dead parasites into animals, they are the best protected. In fact, we also have data. We means not me in my lab, but the whole global malaria community I'm talking about. So there is already data where there is 100% protection with killed parasites. But whether these will be approved and such vaccines will become available it's a very difficult thing to predict.
Okay. Thank you, sir. So since majority of the people that are watching are UG medical students, is there any message that you would like to give to them, or any advice you'd give, common mistakes you see students making in research? Anything you'd like to say?
No, I think I don't want to talk about mistakes, because I think everybody's mistake is their own experience. I think people will learn from experience. That is part and parcel of your learning process. You have to make some mistakes, but the mistake should not be a very costly mistake, of course. You learn from your mistakes. So that being said, I think all of you students, like especially medical students, you should think about couple of things. One is, particularly in India, I see that there is the integration of medical education with cutting edge research is not I would say on par with other countries, like advanced Western countries and things like that. So if you go to the US, or any other European country, you would see every medical college having extensive research laboratories where they're doing a lot of, and they're not only medical students but even PhD students doing, and many of the doctors even do PhD. So you don't know about MD, PhD programs and things like that. I think when medical professionals do a lot more research, more innovation will come. Right now, I think India is very good in giving quality healthcare, but whether we are advancing in terms of innovation in healthcare, I'm not sure, because many of the advanced technology actually come from other countries, and then we implement them very well here. Of course, in terms of hospital treatment, we have done a lot of innovation. I'm not doubting that at all. But I'm just talking about basic fundamental research in terms of understanding cancer at the molecular level, or infectious disease at the molecular level. Even in case of this COVID, India has now, I think it is number 4 or number 3, I don't know exactly, but and we should be spending a lot more time and effort and resources in doing active research into that. From my perspective, I see a lot of research institutes getting into COVID research, but how about the medical, professionals? I guess they are also, their hands are full by treating patients. I don't deny that, but at some time, I think we have to think. There is AIMS New Delhi doing some research on COVID, and there are a few other hospitals I know where research is going on, but we need maybe 100 more hospitals doing this. So as students need to think, because you are going to be the future doctors, you are going to be running hospitals, you are going to be running medical colleges. You need to think how at the policy level things can change, or what kind of new programs can be initiated to encourage more fundamental research in medical sciences.
Okay, thank you, sir. So with that, we come to the end of this session, and I would like to thank sir for taking the time to talk to us today. Thank you very much. And for everybody that's watching, please stay tuned for more videos like this. Thank you so much for your time. Take care.
Thanks so much.
Yeah. Bye.